In this video, I will present the WHEAT Genomics Scientific Literature Knowledge Graph, SLKG for short, as well as the user interfaces that we have developed to query and explore this knowledge graph. This work was carried out in the context of the D2CAP project. So to start, let me just illustrate the motivation of this work. In many research domains, the pace of article publication is skyrocketing. For instance, if I look up the keywords WHEAT genomics on PubMed, I will get more than 15,000 results. This is a huge wealth of knowledge, but of course, aggregating and making sense of the knowledge from so many research articles is barely possible for humans. So we need help. Let's take this article. If you look at the abstract, we can identify mentions of concepts of interest in the WHEAT genomics domain, such as genes, life traits, taxa, as well as other types of concepts that do not show in this example, such as generic markers and plant varieties. So to explore and eventually exploit this knowledge throughout thousands of articles, we need to represent the knowledge in a machine-readable format. So to do so, in a first step, we use the tool called AlvisNLP to identify the mentions of such concepts in the abstract of each article. We call these mentions named entities. But only identifying mentions of genes, traits, and taxa is not enough. We also need to link these mentions with reference vocabularies so that we have a unique way of naming things. So in this example, the tool has identified the mentions of two genes, a trait, and a taxon. And it will also link the trait to the WHEAT trait and phenotype ontology, WTO, and it will link the taxon to the NCBI taxonomy, such that whether articles mention common wheat or Triticum aestivum or Triticum vulgare, they are all connected by the fact that they mention the very same concept in the NCBI taxonomy, whatever the syntax, whatever the language. And this works the same way for the traits and the genes and so on. So the next step is to turn all of these mentions of named entities into a knowledge graph. Briefly, if you are familiar with RDF knowledge graphs, here is the model. We use the Web Annotations Ontology, which is a W3C standard, and we represent each mention of a named entity as an annotation. The target of the annotation represents the specific piece of text that is being annotated. Here, this is leaf rust resistance in the abstract. And the body of the annotation is the URI of the concept that is being represented. Here, this is the concept of the leaf rust resistance within the WHEAT trait ontology. This transformation is carried out by a tool called MorphXR2ML that is able to transform heterogeneous data into RDF. In addition to that, we retrieve from the PubMed API the article's metadata, including the authors, dates, identifiers, title, and so on. And we represent these metadata with common vocabularies such as Deblencore, bibliographic ontology, and Fabio. And this time, the transformation is carried out with an architecture that we call the Sparkle microservices. So once we have completed these steps, we obtain a knowledge graph that contains over 1,200,000 triples, almost 8,500 articles, with 88,000 mentions of named entities. And these named entities are linked with over 4,300 unique entities. Okay, so now let's move on with a demonstration of the user interfaces that we have set up to query and explore this knowledge graph. This first interface allows to search articles by the named entities that they mention. The user starts by typing a few letters in the search field, for instance, rust. And the autocompletion proposes the matched gene names among the genes, trait, taxa, genetic markers, and plant varieties. Let's say I select rust resistance, and let's assume I study the relationship between this trait and a specific gene. Well, I just need to type in the name of the gene, let's say LR34. I select it and hit the search button. In the first section of the results, I get the articles that mention exactly those two named entities, rust resistance and LR34. So using the icon on the right of each article, I can open a new page with the details of each article. Here I get the metadata. And in particular, I have this part that highlights the named entities that have been identified in the abstract. Here I can notice rest resistance, which is the trait that I have entered. I can notice LR34, which is the gene I was looking for. But there are also other entities such as the wheat, Triticum aestivum. And if I click on the named entity itself, I'm being redirected to the page of NCBI or the page of WTO. Okay, so let's close this one and let's continue. The second result section down here adds the articles that mention either the named entities or any of their subconcepts. Here you can notice resistance to stem rust, which is not exactly what I entered. I entered rust resistance. Rust resistance is a trait. Resistance to stem rust is a phenotype related to the trait. So doing so, I'm looking in more specific concepts in addition to the one that I have entered precisely. And this way, I can search articles while traversing the hierarchy of the reference vocabularies to which the named entities are linked. And therefore, I leverage the formal knowledge that these vocabularies provide. So this is fine, but here I need to know in advance what I'm looking for. I'm basically trying to verify an hypothesis I may have in head. But I may also want to explore the corpus of articles to discover relationships that I may not yet know about. For this, I can use the second interface, LDVs, that relies on the MGExplore library. Here, several types of queries are predefined. For instance, I can select the second one that looks for the co-occurrence of mentions of trait or phenotype and any kind of gene. Here, I enter my research, which is resistance to rust, and I hit the run button. The result here is a graph. Each node represents either a trait or phenotype or a gene. The green nodes are the traits or phenotypes. You can see the resistance to rust, which is the one I have entered, but also resistance to leaf rust, resistance to stripe rust, and stem rust, which are the phenotypes related to the resistance to rust. The orange nodes are genes. A link between a trait or phenotype and a gene means that they are mentioned together in at least one article. If I choose that the size of the nodes will be representing the number of papers in which they are mentioned, I can see that some of the genes are bigger than the other ones. This one in particular is the LR34 I was using in my previous example. And I can see that this one is mentioned in 60 publications, and it is related with the 4 traits or phenotypes. To find out more details, I can use different visualization techniques. For instance, the egocentric view places the gene at the center, the node I have selected, and the blue bars give me an idea of how many articles do mention this gene together with this trait or phenotype. Now, here I can see that 34 papers do mention the resistance to leaf rust and the LR34 gene. Now, if I right-click again, I can have the distributions of those papers through time. I can also have the list of papers that are concerned. And here, from this list, I can link directly to the user interfaces I was presenting at the beginning, which presents the metadata and the abstract, where I can highlight the named entities. Now, note that this interface is very flexible. The nodes that are represented here can be anything in the graph, not only the named entities. Let's take another example. I take the first query here, and I hit the run button. In this example, the nodes of our authors. When there is a link between two nodes, that means those two authors have at least one publication in common. They are co-writers, co-authors. And again, you can get to the more advanced visualization techniques. Here, there is another one that I have not mentioned yet, which is the cluster view. Again, I can use the egocentric view. And again, I can see the list of publications of these people and so on and so on. So as you can see, this is extremely flexible. If you would like to know more about this knowledge graph, here is all the information about the dataset published under an open license, the public SPARQL endpoint, the open source repository, the user interfaces, and the publication that describes this work. Thank you for watching.